everyone. I'm Riddhi. I'm a graduate student at the Earth Life Science Institute. In our study, we have tried to understand scaling of protein function across the tree of life and the mechanisms that might have led to the species diversity on the tree of life right here. So we study scaling using power laws, and power laws are found everywhere. If we consider the number of web hits on a web page in a given period of time, or the earthquake magnitude in an area over a given period of time, many natural and man-made processes follow power laws. So how can we how can this help us understand some concepts in biology? For that, let's consider a Lego set and consider the unique pieces in relation to the total pieces in the Lego set. When we plot this on a log-log scale, we see the larger Lego sets use more unique piece types, but they progressively go on using lesser additional piece types. So they're becoming more efficient, which means the larger sets are using the same pieces the smaller sets are using, but in more efficient and more complex ways. So what we're observing in these plots is a scaling relationship. And when we observe a scaling relationship, we could say that maybe there's a set of rules that's sort of governing the way something is working. So here's a power law equation, one quantity varying as a power law of the other. And when we plot this on a log-log scale, we get a straight line with the slope alpha. So previous studies have shown that genes in a specific functional category scale as a power law of the total number of genes in a genome. So for example, transcription regulation is almost quadratically scaling, which means if the genome doubles in size, the genes in this specific category are going to quadruple. So we have tried to include an expanded taxonomy in our study, and for that we use the EGNOG database. So after power law fitting, we saw different trends in our data for the smaller and the larger genomes. So we carried out piecewise regression to give justice to the different patterns in scaling observed in the plots. So for example, we wanted to capture the slope variability for the smaller and the larger genome sizes. So for example, in category M, which is cell wall and cell membrane proteins, so the x-axis has the total protein annotations and the y-axis has the category annotations for that specific category. So for the smaller genomes, we can see the proteins are scaling fast, which means as the genome size is increasing, they are incorporating more and more proteins faster than the larger genomes, because after a statistically detected breakpoint, the scaling slows down. And we can see a similar trend in category T. So we saw this trend in most of the breakpoints that were supported in bacteria. Interestingly, we observed an opposite trend in archaea. The scaling is slow in the start, but fastens up after the statistically detected breakpoint. So a lot of categories were common between archaea and bacteria, but there were some categories that were exclusively present either in archaea or bacteria. And it's also interesting to observe these differences in scaling pattern before and after the breakpoint in archaea and bacteria. So we thought maybe these differences in scaling patterns were caused by phyla-specific scaling. So we broke these domains down into specific phyla and found great variation in all the phyla for all the categories. So for example, we have category H here, which is coenzyme transport proteins, which also happens to be the most variable across all the phyla. So we thought maybe this phyla-specific scaling is causing the positioning of the breakpoints that we observed previously. So I placed these breakpoints on the total protein annotations to see if there's any specific pattern. But we can see these individual phyla are spanning the breakpoints, and there is no specific preference for the phylas to be present on either sides of the breakpoints. So maybe taxonomy is not causing the positioning of these breakpoints, and maybe there are some other factors like physiological or environmental factors that are causing these breakpoints. So we were also interested in these groups, CPR and DPAN. So these groups have extremely small genomes and they lack major metabolic pathways. So we compared them with eukaryotes, unicellular eukaryotes and Asgard archaea. So for some categories, we can see the scaling is very similar for category O, but for some categories, the scaling is very different, like in category C, which goes on to show there are different ways in which an organism can adapt while growing in their genome sizes. I've just discussed a few key results in my talk, so if you want to discuss, I would be interested. Please come by and stop at panel 2 for the poster. Thank you.